Hi everyone, this is Alex, and today's pharmacology topic is going to be the basic mechanisms of drug actions. So the first one that we're going to start with is binding to endogenous proteins. Now there are several subtypes or ways in the body in which this can occur. The first one is through receptors. Now a receptor mediates the effects of neurotransmitters, hormones, and autocoids. The drug that binds to a receptor is called, in this case, a ligand. Okay, so the several subtypes of drug or types of ligand that bind to this receptor is firstly an agonist, and this produces an effect. So for example, acetylcholine binds to the muscarinic receptor. Then we have antagonists which inhibit the effect. For example, atropine. And these bind also to muscarinic receptors. Allosteric modulators, which bind to another site and change the conformation of receptor function. For example, benzodiazepines, they modulate the chloride GABA-gated channels. So what would happen is instead of binding here, they would bind here, and they would change the conformation of this receptor binding site from this to, let's say, this. And therefore, the original ligands or endogenous substances cannot bind here. And lastly, an inverse agonist. This decreases the level of activation of the receptor. So the next subtype that drugs use to bind to the proteins are ion channels. These are several subunits that bind together to form a central pore, as you can see here, with a concentration gradient going in and going out that controls the ion influx. Now there are several subtypes of ion channels, the first of which is a voltage-gated ion channel, and this occurs when membrane depolarization opens the channel. Next we have a channel blocker, and this is when a drug plugs the pore, so it would block this entrance here. For example, local anesthetics. Then you have channel modulators, which have specific site binding and conformational changes. So it bind to a site here and then cause this pore to change shape, preventing the ions from going through. For example, aldosterone. The third subtype that drugs use to bind to proteins are enzymes. These catalyze the endogenous reactions. There are several types of effect that drugs have on enzymes. For example, inhibition, which is reversible, irreversible, competitive, and non-competitive. I'm going to go into more detail on these subtypes in another lecture. False substrates, which are structurally similar to the substrate. So what happens is, for example, if this was not the real substrate, which would catalyze a reaction by binding to this enzyme, because it's the same shape, it would bind here, but then it would not be released here. It would block the enzyme's binding site, and the enzyme would not work anymore. if it did get released, it would make a false product. So this is the other way. False product. For example, methyl-dopa instead of L-dopa. Then lastly, we have the enzyme activator. This increases the reaction velocity and the catalytic capacity to modify the enzyme. For example, nitroglycerin. The last subtype, number 4, which drugs use are transporters. These facilitate the movement of substrates across the membranes, as you can see here, from extracellular to intercellular. There are 2 subtypes of drugs that act upon these transporters: blockers, which inhibit the transportation, or alternative substrates. These compete for the binding site. A blocker, for example, would be cocaine, that inhibits uptake 1. And then an alternative substrate would be amphetamine, which competes for the binding site, again on uptake 1. So now I've finished with the first mechanism, which was binding to endogenous proteins. Now I'm going to tell you about the other mechanisms. So number 2 would be maintaining the structural integrity of the cell. And my example will be colchicine. Which acts on tubulin. Also, the neutralization of endogenous materials. For example, protamine with heparin. It's used to cure an overdose of heparin. So it basically eliminates or neutralizes the heparin. And lastly, osmotic regulation. For example, laxatives or diuretics.